water carries things through our body. It will carry those metabolites by taking them from our muscles to our kidneys so they can get rid of them. The oxygen will also get carried from our lungs to the rest of our body. Some of it will dissolve in the water itself. Most of it will go into the red blood cells, which get carried by the plasma, which is mostly water. All the minerals in our body, the calcium, the phosphorus, the sodium, the potassium, everything, is going to dissolve in water. And so that water will help move it around. Water is what makes our stool soft enough that we don't get constipated. Not only does it lubricate joints, but it provides some cushioning around our organs and tissues. Remember our serous membranes. There is that serous fluid in the membrane. That's mostly water. And then it also allows us to keep certain parts of our body wet. It'd be very hard for me to be talking to you if my mouth wasn't wet, right? So water plays a lot of different roles in our body. Our inorganic compounds are basically going to fall into three categories. We have acids, bases, and salts. An acid is a substance that when you break it apart, it's going to give us hydrogen ions. Essentially, an acid is going to be an ionic compound where the metal is hydrogen. If the positive ion, the metal in an ionic compound, is hydrogen, it's going to be an acid. Hydrochloric acid, your stomach acid, is HCl. This is an ionic compound. Hydrogen is our metal. It's on the left-hand side of the periodic table. And so this is going to have a positive charge and a negative charge. Something that has hydrogen in an ionic compound is going to be an acid. A base has an OH in it. OH is called hydroxide. A base is going to make hydroxide when you dissolve it in water. And so an example of that is NaOH. If you take an ionic compound and you put it into water, the ions break apart. So if I put HCl in the water, I get H plus and Cl minus. They float apart from each other. So that says if I make H plus, I'm an acid. If I put this in water, I get sodium and I get hydroxide. I just made hydroxide. So that is a base. Acids and bases react with each other. The hydrogen from this acid will react with that hydroxide from that base. So then I have H plus OH. Put it together, what do you get? H2O. I now have two hydrogens and one oxygen. So acids and bases essentially are opposites of each other. They're going to react, they're going to cancel each other out, and when they do that, they're going to make water. A salt is an ionic compound that does not contain hydrogen or hydroxide. So an ionic compound can be an acid, a base, or a salt. If it has H plus, it's an acid. If it has OH minus, it's a base. If it has neither, it's called a salt. We use the term salt for sodium chloride all the time, table salt, right? But in reality, table salt is just an example of a salt. KCl, potassium chloride, is a salt. If you had magnesium sulfide, that is a salt also. Okay? It's an ionic compound. Magnesium is a metal. Magnesium is on the left-hand side. And so this is ionic, but it's not a base, not an acid, so it is a salt. All of them are what we call electrolytes. An electrolyte is something that when you dissolve it in water, will conduct electricity. For something to conduct electricity, you have to have ion. You have to have charges. Ionic compounds have charges. That's what makes them ionic compounds. And so when you dissolve them in water, they will conduct electricity. So here's hydrochloric acid, HCl. Put it in the water, and again it breaks apart into a hydrogen ion and a chlorine ion. You take potassium hydroxide, there's an OH right there. Put it in the water, you're going to get a potassium ion and an OH ion. What is this? Is this an acid, base, or a solid? It's a base. An OH means it's a base. Potassium chloride, KCl, gives us K and Cl, and that's a salt. It does not have an H plus, it does not have an OH minus, so everything else is a salt. We measure how acidic or basic a solution is using a pH scale. pH scale is going to tell us whether there is more hydrogen ions or if there are more hydroxide ions. You're not going to have to calculate pH, but this is how you would do it. You would take the hydrogen ion concentration, so this H3O plus is the same as just H plus. In chemistry we use this is actually more correct. And you would take the negative log of it, okay? You don't have to do it. When you do that, you get this scale. Most people think the pH scale goes from 1 to 14. In real life, most things will fall between 1 and 14, but it actually goes infinite in both directions. You can have a negative pH, you can have a pH of 1,000,000. pH of 7 is neutral. If you have a pH that is less than 7, mathematically, that means that this is going to be a large number, which means you're acidic. What you need to remember is that a pH less than 7 is acidic. A pH over 7 or above 7 is basic. If you are basic, that means that you have more hydroxide than you have H plus. If you are acidic, that means you have more acid than you have base. Our body is a little bit basic. Blood pH is 7.4. So most things in our body are very slightly basic. Some of the other things like milk and urine are acidic though. Things that we use in lab or things that we use to clean can be very acidic. Lemon juice is down here, vinegar. Stomach acid is very acidic. That's hydrochloric acid. It's a very strong acid. It's a pH of 1. We don't have many things in our body that are strongly basic. To get very hard, strong base, we need things like ammonia and sodium hydroxide, which would be something we would use in the lab, not even something you would have at home. Remember, our pH of our blood is about 7.4 or 7.35. If you become more basic, we say that you're suffering from alkalosis. If you become more acidic, we say you're suffering from acidosis. If you go too far either way though, you die. The enzymes in our body only work at a very narrow range of pH. They have been designed to work at a specific pH. And if you get outside of that range, the enzymes stop working. And the enzymes in our body accomplish everything. If they stop working, our body stops working. So normally, our pH is very close to 7, which means we have about the same amount of acid in base. We have systems in our body, though, that prevent these large changes. We want to keep our pH very close to 7.35, and the system that prevents those large changes are called buffers. Buffers work to take a strong acid and convert it to a weak acid. They take a strong base and convert it to a weak base. We haven't talked about these terms. We don't need to talk about what they really mean. Strong and weak are pretty self-explanatory for what we need to cover. If you drink something that's a very strong acid, our body can take that strong acid, convert it into a different acid that's not as strong. It'll be weak. And so a weak acid is not going to affect our pH nearly as much as a strong acid. Let's take our break now. So let's come back at 25 after on that.
before covered all of it, but not in the back. But it's more difficult because it's great. So I think generally 24 after going into some of the details, it's 25 seconds.
Whoever does that is an easy question. You can provide them that primary buffer system we're going to talk about is what? So if you drink it, you're going to completely burn everything down, you know, whether you can neutralize that or not. So we're going to be part of that lecture. It's however you take notes, how to be recording. Right, so whatever we don't cover will be our lecture next week. So however, how you take notes, on a tablet or on paper? So if you would normally take notes on the tablet, then I would just take the tablet. Take notes. Just pretend it's a lecture. We're just sitting there. That's it. Leave the door open after that. So we said that our body wants to keep a narrow range of pH, right? We said a buffer is a system that allows us to prevent those large changes. Our body has a number of different buffer systems. The primary one, though, is called the bicarbonate buffer system. And so it's kind of a three-part system here. And so here we have arrows pointing in both directions, here and here. And so what this is saying is this H2CO3, which is acid, that's carbonic acid, can be converted into H plus and HCO3. It can also be converted into CO2 and water. So if we take
acid, let's say that we just exercised. When you exercise, you create acid in your muscles. So your muscles just created a bunch of acid, and this is in your bloodstream. It's going to combine with a base in your bloodstream. This is bicarbonate. This is produced by your kidneys. So this is just floating around in your bloodstream, waiting for the acid to come along. The acid gets made, they react, they make carbonic acid, and then carbonic acid is actually going to break apart in the CO2 and water. The CO2 is a gas. We exhale CO2, right? So what this system allows us to do is that we make acid. It gets converted into a different form of acid, and then we make CO2, and we exhale the CO2. So essentially what we're doing is we are exhaling the acid as we make it. We make acid, but it reacts with a base. And so it neutralizes the acid, the pH doesn't change, but we only have so much of this base in our bodies. And so we just can't keep using it up as we make acid. We need some way to get rid of the acid to free the bicarbonate up again. And so we have this two-part process. We'll breathe out CO2. When we do that, our bicarbonate is now free to bind to another hydrogen atom. If this doesn't work, then we get acidosis. And you can see you're not responsible for specifics here. But essentially, if you become acidotic or alkalytic, which is too basic, everything in your body goes haywire. Because everything relies on enzymes, and so everything is going to stop working. There are two other buffers in our body, though. We have the phosphate buffer, which is very similar, but it goes between phosphoric acid and this H2PO4. And hemoglobin itself, its primary role is for oxygen, but it can also bind acid. And so that will help prevent these changes in our blood pH. Okay. That's the end of our inorganic biochemistry. So we had acids, bases, salts, and then the fact that a buffer acts to maintain a normal pH. So organic biochemistry is probably the heaviest stuff here. So in order to talk about organic biochemistry, we have to know what organic chemistry is. And organic chemistry is the chemistry of carbon. But carbon itself has no special talents. Carbon by itself can do very little. And so what we're going to do is we're going to add things onto our carbon. And those things that we add on are called functional groups. They're things that we're going to add to the carbon that serve specific purposes. And depending on what these functional groups are, we're going to make 4 different categories of organic molecules. We have carbohydrates, which are sugars, lipids, which are fats, proteins, and then nucleic acids, which make up our DNA. So carbon on its own has what we call 4 valence electrons. All atoms have a number of electrons, but the only ones that are going to react are the ones that are on the outermost shell. And you don't need to be able to figure out how many valence electrons any atom has, but it's important to realize that carbon has 4, and so it's going to want to make 4 bonds. Whether that's with carbon, or with hydrogen, or with oxygen, or nitrogen, or anything else, it's going to want to make 4 bonds. And it's going to want to make 4 bonds because it wants to fill its outer shell. Each shell can hold 8 electrons. Carbon has 4, and so it needs to make 4 bonds to gain 4 to get to 8. Everything wants 8. Carbon has 4, so it needs to make 4 more bonds. The carbon will bond to itself very easily. And when it does that, it's going to make these very long chains of carbon. You've probably seen molecules that look like that. These are carbon chains. Each corner is a carbon. And that can go forever and ever and ever. And it can branch. You could have things coming off like that. But it's all going to be carbon. And these are all covalent bonds. Here we're only going to be talking about covalent bonds because everything is on the right-hand side of that periodic table. There are going to be no metals here. Primarily, it's going to be hydrogen, oxygen, nitrogen, and sulfur. It can form bonds with other things like silicon, but it's very, very rare in our bodies. And so we're going to imagine this carbon backbone. You have these strands of carbon, but then we can add things onto them. You can add a phosphate. You can add a hydroxide. And phosphates and hydroxides and all these other functional groups will behave differently. And so the arrangement of those functional groups on our carbon skeleton is going to determine the properties of our molecule. Because a carbon backbone by itself does basically nothing. So we have a few functional groups up here. This is an OH. You can add more carbon. So you can add more carbon. You can add an OH. You can add carbon with oxygen, COOH. You can add nitrogen with hydrogen. You can add phosphates, that's phosphorus with oxygen. And they're all going to have different purposes. So this is hydroxyl, that's the OH, methyl, that's the extra carbon, carboxyl is that, amino is that with the nitrogen, and phosphate has the phosphorus. We can also take carbon molecules and we can string them together to make bigger carbon molecules. So a macromolecule is going to be a lot of these smaller molecules strung together end to end. And so you're going to end up with a very large molecule with a very high molecular weight. These are polymers. So a polymer is just a straight chemical term, a term from chemistry, where you take a number of copies of a small molecule that are all identical, and you're going to string them end to end. Each individual part is called a monomer, so mono one, poly multiple. You take a bunch of monomers, string them together to get a polymer or a polymer. Starch is a whole bunch of glucose molecules strung together. It's very big. One starch molecule has about 3,000 glucose molecules strung end to end. The process of putting them end to end is called polymerization. And this is an example. So here we have two monomers. We have the blue monomer and the orange monomer. This is actually straight out of the book. I don't know if they're Gator fan or not. But we have two monomers, and then it's hard to see up here, but that actually has an H, and that has an OH written in there in white inside the blue here. And so if you take the two monomers, stick them together, that H is going to react with that OH to make water, and then the two monomers are going to be stuck together. Two monomers together is called a dimer. You have dimer and then tri for three, trimer, and then polymer. This is called a dehydration synthesis because we took water out. There was H and OH. When we put them together, the water left the compound. So it's a dehydration, and it's called a synthesis because we made something big. If you take small things, make something bigger, that is synthesis. The opposite is called hydrolysis. If we take our dimer and we put water back into it, it's going to break this bond back apart, and now we have H and OH. We took our dimer, we turned it back into monomers. hydro for water, lysis is breaking apart. So we're using water to break apart our dimer to make monomers. That brings us to carbohydrates. Carbohydrates are going to have a generic formula, and they're all just going to be multiples of each other. So this says that we have a general formula of CH2O with a subscript N. And that N is going to be however many carbon atoms are in that carbohydrate. And so a carbohydrate, the simplest carbohydrate is CH2O. If we want to make this bigger though, for every carbon that we add, we have to add 2 hydrogens. And we have to add 1 oxygen. And so I can go C2H4O2. If I want to make it bigger, I can go C3H6O3. Does anybody happen to know the formula for glucose? C6H12O6. That fits this general formula. There's the same number of oxygens as carbons and twice as many hydrogens as carbons. So glucose is a carbohydrate. Oh, it's right there. How come only one person said it? Essentially, we have a 2-to-1 ratio of hydrogen to carbon and a 2-to-1 ratio of hydrogen to oxygen. We're going to have a number of carbohydrates that have names. Pretty much all of them will end with os, which means sweet or sugar in Latin. The saccharine or saccharose, that also means sugar or sweet, if you see that in the name. So we have 3 monosaccharides, which are important in our bodies. So these are going to be our monomers that are our carbohydrates that we can combine together to make big polysaccharides. We have glucose, galactose, and fructose. They are all C6H12O6. The only difference between them are the connections. So we're not going to go through these in detail, but each one is a different one of these monosaccharides. Just the bonds are slightly different connections. So some atoms are connected to others, we're in the other one, it's not. It's probably easiest to see down here, because now we have a 5-membered ring instead of a 6-membered ring. Those are called isomers. An isomer has the same formula, but different connections. We get these monosaccharides by breaking down bigger molecules that we eat. If we eat something, there's only going to be a little bit of these monosaccharides in there. We're usually going to have bigger things like starch that we're going to have to break down in order to get these. So a monosaccharide is one, a disaccharide is 2 of them together. And there are 3 disaccharides that you're responsible for knowing. We have sucrose, which is just normal sugar, and that's a glucose with a fructose. Lactose in your milk is a glucose and a galactose, and maltose is glucose and another glucose. So maltose is very common in wheat products. So you're responsible for knowing these names and also knowing which monosaccharides go into making that disaccharide. An oligosaccharide is a short chain of monosaccharides. You have at least 10, but it's not incredibly long. And where this ends is not going to be a clear-cut definition. Essentially, you're going to have things that are big, that are a reasonable number, and then you're going to have things like starch that are 3,000. Okay? A polysaccharide is longer. So this says at least 10, this says at least 50. If you want to make 50 your cutoff, fine, but nobody's going to argue with you if you say it's 75 or 40. Examples of polysaccharides are glycogen. When we took all the glucose molecules, strung them end to end, we got glycogen. Starch is essentially the same. There's just a very, very minor difference. Although starch is in plants, glycogen is in animals. Glycogen is essentially animal starch. And then there's cellulose. Obviously, animals don't have cellulose either, but that is a giant chain of monosaccharides together to make a giant polysaccharide. This is our glycogen. Again, glycogen can branch. It's not just one big long string. You can get branches coming out. And so this is just zooming in on this region. And so we have glucose, 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 and then they're bound to each other. And so if you took this glycogen and you carried out glycolysis, you could start breaking these off, and as you got them, you'd get energy out of it. Carbohydrates are used for energy, and they're very easy for your body to break down. And so it's very quick. If we need quick energy, number one, we're going to use the monosaccharides that are floating around in our bloodstream. We always have some glucose in our bloodstream, right? And so that's the first thing that's going to get used. If we need more of that glucose, the first thing our body's going to go to is the glycogen. Because if we need to make glucose, glycogen is just a bunch of glucose strung together. So if you chop that up, you have glucose immediately. Other things like lipids and proteins, they're nothing like glucose. So your body's going to have a long, expensive process to convert those back into glucose. So carbohydrates are quick energy. When you digest a carbohydrate, it gets converted to glucose. When you take a carbohydrate in, it gets converted to glucose, and then your body decides what to do with it. Am I going to burn it? Am I going to store it? We can oxidize it. So oxidation is essentially burning the sugar, and we're going to make ATP. ATP is a molecule we'll keep coming back to. It's essentially the energy currency of the cell. If we burn glucose, we get energy in the form of ATP. We can also take these carbohydrates and bind them to other things. You don't have to just stick them to other carbohydrates. If you stick a carbohydrate to a lipid or a fat, you get what's called a glycolipid. These are present on the outside of cells. So they stick out from the cell membrane. The main purpose of these is for signaling other cells what a cell is. It's kind of like a flag sticking up saying, I'm this type of a cell, I'm here doing such and such. You can take a sugar and you can stick it on a protein to get a glycoprotein. Again, these can be on the outside of the cell, but they're also present in our respiratory and digestive tract, primarily where you would find mucus. And then you have what's called proteoglycans. Proteoglycans are very, very similar to glycoproteins. It's just there's more carbohydrate than protein. So it's kind of like sticking a protein onto a big carbohydrate. These can form thick solutions, so you get kind of a gel. And so anything in your body, like the filling of your eye, where it needs to be kind of a thick water solution, is going to have these proteoglycans. And then also the lubrication in our joints. Water itself is not the best lubricant, right? You want something a little thick that'll be a little slipperier. That brings us to the lipids. Lipids, it says, are hydrophobic organic molecules. Organic meaning carbon, hydrophobic meaning they don't like water. And you know that. You know that oil and fat don't dissolve in water. That's what this means. It also says that there's a high ratio of hydrogen to oxygen. So whereas a carbohydrate, for every carbon you add, you have to add an oxygen. A lipid is going to have a lot more hydrogen than oxygen. Here it's 2 to 1. A lipid is going to be a much higher ratio. Lipids have a lot more energy stored in them. That's why when you eat fat, you get a lot more calories out of fat than you get out of a carbohydrate. And there are 5 main types of lipids. There are fatty acids, triglycerides, which we'll see a picture of how those go together. There's phospholipids, which we'll talk about a lot in terms of the cell. And then there are eicosanoids and steroids. So this is a generic picture of a lipid. There's a lot of carbons, a lot of hydrogens, and then there's just a couple oxygens down here. This is a fatty acid. On this end, we have a carbon, an oxygen, and an OH. So it's a big long string of carbons with oxygens on just one end. That's going to be a fatty acid. This group down here is this carboxyl group. The methyl group is just saying that it ends with a carbon. So you've heard of saturated fat and unsaturated fat. A saturated fat means that it can hold no more hydrogen. An unsaturated fat means that it could potentially hold more hydrogen. It can hold more hydrogen if it has double bonds. If I make this bond between these two carbons a double bond, I have to remove a hydrogen off of each one. And so if I take fatty acid with a double bond, I can react it with hydrogen to put hydrogen back onto it. That's what a hydrogenated oil is. They take unsaturated oil, they put hydrogen into it. They make it hydrogenated. They put the hydrogen back in. A polyunsaturated fatty acid has a number of double bonds. So the more double bonds you have, the more hydrogen it's capable of holding. Our body can make most fatty acids that we need. There are a few, though, that we can't. So we have to eat those. We can live without them, we just may not be as healthy as we could possibly be. So we have to eat them in order for our body to have them. A triglyceride is 3 fatty acids glued to one kind of backbone. And so it's 3 fatty acids linked to glycerol. Glycerol, you can sometimes hear the word glycerin. Glycerol and glycerin are the same thing. And we use that dehydration synthesis where you take water out to make it happen, and we break it apart by hydrolysis. If you have a triglyceride at room temperature, and it's an oil, when it's a liquid, we call it an oil. A triglyceride, it's a solid at room temperature, is a fat. That's the only difference between an oil and a fat. It's what state it's at when it's at room temperature. Usually, oils are polyunsaturated, and they usually come from plants. For the most part, fats are saturated, meaning they have as many hydrogens as they can hold, and they usually come from animals. Triglycerides are usually used for energy storage. The fat in our bodies that we use for storage, at least in purpose, is for energy storage, and that is in the form of triglycerides. It also provides insulation and some shock absorption. So if I get punched in the gut, I may be a little safer than some of you more fit people. So a triglyceride, we said there's 3 fatty acids linked to a glycerol backbone. So a glycerol backbone is just 3 carbons. And so what we're going to have is we're going to have 1 fatty acid here, 1 fatty acid there, and 1 fatty acid there.
You may see it also drawn just kind of in a line where they all come down like that. You're essentially taking 3 fatty acids and combining them into one convenient little package by linking them across the top. Next we have phospholipids. They're very similar to our triglyceride, except there is a phosphate or a phosphorus stuck in there. These are found in our cell membrane. When we talk about our cell membrane, we're going to see that it's made up of these phospholipids. What this really causes is this phosphate group is very polar. And so you're going to have a polar end that likes water and a nonpolar end that doesn't like water. And so you get this double membrane of the tails of the phospholipids with each other, and then the phosphate groups on the inside of the cell, where the cytoplasm is, and then on the outside of the cell, where there's a bunch of water. The fact that it has these two ends, we call it amphipathic. Okay, like amphibious, water and land, amphipathic. It means that the tails away from the hydrogen, away from the phosphate, so down here are going to be hydrophobic, and then the phosphate on the head is going to be hydrophilic. It's going to want to be near water. This is a term that's going to come up occasionally. This is an eicosanoid. These are kind of a very specific type of molecule, and they always have 20 carbons, and they always get made from a molecule called arachidonic acid, which is this. These get used similarly to hormones. They get made in one part of the body, and they travel throughout the body and tell other organs what's going on. This includes prostaglandins. Prostaglandins have important roles in things like inflammation and blood clotting. Okay? That's why if you take an aspirin, it prevents inflammation and it prevents blood clotting. It affects this whole process. If you take fish oil, that will also affect this process. A steroid is a lipid that has 17 carbons and is going to be 4 rings in the molecule. Certainly the best known of these steroids is cholesterol. Cholesterol, it says, is the parent steroid from which all other steroids are made. So we have a bunch of different steroids in our body, but our body makes cholesterol and then converts them to all of the different ones. It's important for nervous system function. It's important for keeping our cell membranes from breaking. It actually provides some flexibility in our membranes. Otherwise, we didn't have it. When you bent the cell membrane, it would just snap. We get a little bit of our cholesterol from what we eat, but the vast majority of it comes from our body. We make the cholesterol. That's why how much cholesterol we eat isn't as important as they said it was in the 80s. Back in the 80s, it was very important. If you had high cholesterol, you were supposed to limit and eat as little cholesterol as possible. Now, they tell you, sure, eat as little as possible, but in reality, we need to be controlling how much you make, which is why we take a lot of things like Lipitor. That's going to stop us from making as much. Things like cortisol and estrogen, testosterone, those are also steroids. Then we have proteins. A protein is another polymer, but it's made up of amino acids. An amino acid has 4 parts. There's an amino group, that's the amino. There's a carboxyl group, which is the acid part. And then there is an R group. All amino acids have the amino and the carboxyl same. The only difference is what is this R group. And there are 20 different R groups.
And so if there are 20 different R groups, we get 20 different amino acids. If you're going to make a protein, you have 20 different building blocks. So all of the proteins in your body are different combinations of those 20 amino acids. And what that order is and how long they are is going to determine the structure and function of that protein. And the properties of each amino acid is solely determined by this R group, because this is the same in all of them. The only thing that's different is the R group. So these are 4 amino acids. You don't need to know the names of all the amino acids, but you can see that this part here is the same in all 4, right? So this down here, that these, for some reason that OH is not showing up, but this is the acid part. The nitrogen part up top is the amino part, and then this is the R group. So there are 4 different amino acids here with 4 different R groups, which makes them all behave slightly differently. So you combine them in different orders, and you can make any type of protein that you want to make. We now know enough about these amino acids and how they behave that people can actually say, I want to make an enzyme that does this. I need to take these amino acids, put them in this order, and there I go, I have an enzyme that can do what I want it to do. A peptide is if you take multiple amino acids and combine them together. Amino acids are held together by what are called peptide bonds. A peptide bond is a type of a covalent bond, but it's the specific type of covalent bond that holds amino acids together. It takes the amino, the N part of an amino acid, and connects it to the carboxyl or the oxygen part of the next one. So similar to this, you end up with a chain. In the chain, you're going to alternate carbon and nitrogen. Then these R groups are going to stick up and down out of our chain. Our peptides are going to be named based on how many amino acids we go in, that we put into it. So a dipeptide is 2, tri is 3. Oligo, again, has quite a few, but not a ton. A polypeptide has more than an oligo, but a protein has a ton. Proteins are big. Okay? 50 is going to be a very small protein. Most proteins in your body is going to be much larger than 50. So here we have 2 amino acids. We're going to combine them together to make a dipeptide. The oxygen part on this one is going to bond to the nitrogen part on that one, and now they're linked. And that linkage is our peptide bond. Yes, yeah, because we got rid of this OH and we got rid of one of these hydrogens, so yes, it's a dehydration synthesis again. So depending on that order is going to determine the shape of our protein. And the shape of a protein is called its conformation. Conformation with an O, not confirmation like, yes, you're right. Okay? It's conformation. It's a 3-dimensional shape. Remember, it's real life, so it's a 3-dimensional shape. And its shape determines its function. If you take a protein and you break its shape and you change its shape, it's not going to function anymore. Some proteins can reversibly change conformation, and so their function can change. They can go from one function to another function. They can have one function that can shut off, so it goes from functional to non-functional, but it's reversible, so you can turn it back on. But in some cases, they can change permanently. If you take an egg and you cook that egg, you are permanently changing its structure, its shape. You unfold it, and it can't go back. If you have something like a membrane in our cell channel in our cell membrane, it opens and closes. So depending on the shape, it will allow water or something else through it. and then you shut it off, and it changes shape again, and it closes. So it will shut that water off from going through. Denaturation is when you destroy the function by completely ruining its structure. When you heat that egg, you thought you were cooking it, but you're actually just denaturing the protein. You're denaturing the egg protein. You're also denaturing the proteins in whatever bacteria might be in there, killing the bacteria. That's what you're doing when you're cooking things. You're denaturing the protein. You can do it with heat. You can also do it with a large pH change. Remember I said enzymes works especially well at physiological pH of 7.4. As you change the pH, their shape changes. If you change the pH a lot, the shape is going to change a lot, and they're not going to function very well. We talk about protein structure at 4 levels. There's the primary, secondary, tertiary, and quaternary structure. The primary structure is just the order of amino acids. It's serine, valine, alanine, phenylalanine, something like that. Okay? It's just what order the amino acids are arranged in. That is the primary structure. There are certain little repeated sections that, based on the directions of the bonds in those amino acids, that you'll get what's called a secondary structure. You can either get something that wants to coil, so depending on the sequence of amino acids you put together, sometimes they want to make a little spring shape. Other times you get what's called a beta sheet.